right? So we can see that also here. We can see the monosomy taking place here, and then we can also see trisomy taking place here, and only monosomy taking place there. So we can see the monosomy is represented by the green arrows and the trisomy is represented by the gold arrows, right? Whereas here, there's nothing. Why? Because this would be your meiosis 2 and this would be your meiosis 1. Now, here we are also shown what happens when the abnormal gamete mates with a normal gamete. I'm just going to erase what we already have there so that we can see what happens properly. Right, so over here, we will have this cell that does not have anything, and then it comes into contact. Let's say this is an ovum, and then there we have the haploid sperm cells. Then these would be the resulting offspring. As you can see, this one only has chromosomes from the father as well as this one, and also here there's trisomy because of the non-disjunction that failed to take place. And then here another abnormal cell that combines with the normal sperm cell. We can see here these are normal, and we can see the diploid zygotes. However, here we can see trisomy, and here we can see monosomy. So what you would explain if a question is on how an abnormal zygote is formed, you would mention that the abnormal cell with an extra chromosome combines with a normal sperm cell with a normal number of chromosomes, a haploid gamete combine, and this is what would then result, right? So when these abnormal gametes undergo fertilization with normal gametes, then we can have genetic disorders. We can also have an abnormal gamete undergoing fertilization with another abnormal gamete, which would also lead to another disorder, right? So non-disjunction can lead to aneuploidy, or it can lead to polyploidy, right? Okay, so when we are looking at the condition, we would be looking at a condition whereby one gamete has 2 copies of the same chromosome, meaning that the chromosomes have failed to separate. Therefore, now we have 2 copies of the same thing, while the other gamete will not have a copy of that specific chromosome, right? And then if the gamete with an extra chromosome is then to fuse with a normal one, it means that we will see a zygote with an extra chromosome. Now, which also then means that we are looking at trisomy. Remember, we said that trisomy 21 would be the one that leads to Down syndrome, but trisomy anywhere else would lead to other disorders, and those will not be Down syndrome. Make sure that you remember that. Down syndrome only results from trisomy on chromosome pair number 21. Right. So if the gamete with no copy of chromosomes fuse with another gamete that is normal, then that zygote will have 1 less chromosome. It means that we are looking at monosomy. We can say that the zygote is monosomic, right? This means that the one that has only one it is the one that has only 1 copy instead of 2 copies. So the Down syndrome is brought about by aneuploidy. But remember, it's brought about by aneuploidy on chromosome pair number 21, whereby there is an extra chromosome. It's not brought about by monosomy, right? So this would be something that can potentially have Down syndrome because this is trisomy. But if it is on chromosome 21, then we can say it is Down syndrome. Make sure that you remember that part. This can also occur in meiosis 2, whereby we can end up with chromosomes that have an extra chromosome and cells that have 1 less chromosome. And then also remember that offspring that has trisomy, that offspring will then have a combination of chromosomes that are extra. Instead of 46 chromosomes, that zygote would end up with 47 chromosomes, which is something that you would see when you are looking at a karyotype. Here is a karyotype of an individual with Down syndrome. So on chromosome pair 21, we would see that there's an extra chromosome. We can see that it's even highlighted with a different color just to show that there's something different happening there. So that is the extra chromosome. So questions that you can be asked here: explain why we say this individual has Down syndrome, for example. Then you would say this person has an extra chromosome on chromosome pair number 21, right? Then you can also be asked, is this individual a male or a female? Then you can say this individual is a female. Where do you look to find out for that? You look on chromosome pair number 23. This is a female. Why do you say it is a female? Because they have two X chromosomes on pair number 23, right? So they have the same chromosomes on pair number 23. You can also be asked, how many chromosomes does this individual have? Then you check everywhere else to see if there isn't any other extra chromosome. Then you can say that they have 47 chromosomes because of the extra chromosome there. Okay. The characteristics of individuals with Down syndrome, we can see them by having mental retardation, some hearing loss, some heart defects as well. They also have decreased muscle tone, as well as upwardly slanting eyes. They have a small mouth and a small nose. They have abnormal ear shape, and they have a depressed nasal bridge. So their nasal bridge is not defined, right? And you must also note that there is no cure for Down syndrome. However, some of the symptoms can be treated, but the individual will not be cured of the Down syndrome. So there is a diagram that is indicating some of the physical features of that individual with Down syndrome. Right. So this disorder can also be detected a bit early if the pregnant mother goes through some tests, such as amniocentesis, whereby some fetal cells are removed and they are analyzed to see if there are any abnormalities within the cells of that growing zygote. Right, let's have a look at our second activity. Already when we look at this, we can see that the two cells do not have any chromosomes, right? Then it already tells us that it could mean that non-disjunction has taken place. The diagrams below represent the distribution of chromosome pair 21 as it appears in gametes at the end of meiosis 2 in a human male. Like I said earlier, read the information. Now we already know that we are looking at a male. Now we already know that we are looking at this happening on chromosome pair 21. If the question now says, what disorder will this result in? You would say Down syndrome. However, if it says on chromosome pair number 4, you cannot say Down syndrome, because Down syndrome is not caused by any number of chromosomes missing or extra on chromosome pair number 4, but only on 21. Now, I'm going to give you just a few minutes to go through the questions. Just attempt to answer the questions, and then we will come back and have a look at the questions together so that you can correct yourselves and make some changes to your answers. All right, let's have a look at 2.1.1. It says, explain why the gametes represented in diagrams C and D do not have any chromosomes. What causes cells to not have chromosomes? Right? Already we know that this means that non-disjunction has taken place during anaphase, right? So this is during anaphase 1. Why is it during anaphase 1? This is because there are two cells that are actually empty, whereas if it took place during anaphase 2, we would not have any cells that are empty. We would experience non-disjunction in one of the two resulting cells at the end of meiosis. But now, because the mistake happened right at the top, it means that all the cells are going to be affected. So this shows that it is during anaphase 1. So you score a mark for mentioning when it has taken place, right? And then 2 chromosomes moved to one pole. Now you are explaining exactly what happened when this non-disjunction was happening. 2 chromosomes were pulled to the same pole and none moved to the other pole, which is why we have what we are seeing here, which means that normally what we would expect to see here would have been one chromosome in cell C and the other chromosome in cell D, which would mean that a normal gamete would have 111 chromosomes each. However, in this case, that will not be the case because of non-disjunction. 2.1.2. If gamete A, which has 2 chromosomes, is involved in fertilization, describe how this may result in Down syndrome. So, gamete A looks like this. But if gamete A was normal, this is abnormal gamete A, right? If gamete A was normal, it would have 1 chromosome, right? And then if it mates with a normal cell, it would have another one, and then we would have 2. However, because gamete A is abnormal, it will mate with a normal gamete, and then we would have 2n plus 1. 2n plus 1. So we would have the normal number of chromosomes plus 1 extra chromosome. So that is how it would end up in Down syndrome. So you would mention that the normal gamete would have the haploid number of chromosomes, whereas gamete A would have an extra chromosome, whereby when these combine, the abnormal one and the normal one combine, they would result in an offspring that has an extra chromosome, and that would be trisomy, if this is on chromosome number 21. Gamete A will have 24 chromosomes, or gamete A will have an extra chromosome. And when it fertilizes a normal ovum. Why do we say a normal ovum? Because we were already told that that is in a male, meaning that this abnormal gamete is a sperm cell, therefore it will fertilize an ovum, right? Make sure you mention that it is a mark for that. The zygote will have 3 chromosomes at position 21, or the zygote will have 47 chromosomes. 2.1.3. Due to the process of crossing over, the chromosomes in diagrams A and B appear different to each other. As you saw with the shading, they had different colors. Let's just quickly go back. We can see that this one is plain black, this one is plain white. They are different from each other. This one is 75% white, 25% black. This one is 75% black and 25% white. None of the chromosomes are the same, right? So it says, due to the process of crossing over, the chromosomes are different. A and B appear to be different from each other. Identify the phase of meiosis during which crossing over occurs. Remember, crossing over takes place during prophase 1. If you do not say 1, you lose that mark. B, describe the events during crossing over. Here you must describe what happens during crossing over. It means you now know the importance of meiosis, the importance of crossing over happening, and you know how this crossing over happens. So you're going to describe that the homologous chromosomes are going to lie next to each other. As they lie next to each other, the chromatids of each homolog are going to overlap. As these chromatids overlap, they are going to exchange genetic material. After that has happened, the chromatids are going to separate. When the chromatids separate, each chromatid would now have genetic material from the chromatid of the homologous chromosome, and therefore the 2 chromosomes will now be different. Right, so adjacent chromatids of homologous chromosomes cross, or they overlap, right? At a point called chiasma. Never forget to mention that that point is called chiasma. Remember, if we have 2 chromosomes like this, when they are overlapping here, this point here is called chiasma, right? There is an exchange of DNA segments or genetic material. When that happens, genetic material is going to be exchanged. C, explain the significance of crossing over in natural selection. Now, when you talk about natural selection, remember we are moving on to evolution. However, we know that genetic variation is very important in evolution. Why? Because if there's a great deal of variation, which this great deal of variation is introduced by crossing over, then it means that we're going to have we're going to have organisms that are different from each other. The organisms that show a great deal of variation, even though they are of the same species, which means that we will have those that have favorable characteristics and those that do not have the favorable characteristics. Those with the favorable characteristics would survive, while those without the favorable characteristics will not survive, they will die. Then the ones with the favorable characteristics will survive and pass on the alleles for the favorable characteristics, and that would lead to the species with the favorable characteristics increasing and surviving. So crossing over introduces genetic variation, which results in favorable characteristics, right? That would ensure a better chance of survival when the environmental conditions change. Or because it introduces genetic variation, it may result in those that have unfavorable characteristics, which reduces the chances of survival when the conditions change. So you can mention favorable characteristics, you can mention unfavorable characteristics, or you can even mention both. Okay. Next activity is activity 3, which was taken from question 2.4. It says study the following representation of phases during cell division. We have A, B, C, D, and E. Let's just try to identify these phases before we move on to the questions. Right. So here we can see that we have a telophase. This is telophase 1 because the chromosomes are still full chromosomes or replicated chromosomes. So this is telophase 1. And then at B, we can see that it is anaphase, and it is also anaphase 1 because full chromosomes or whole chromosomes are being pulled to the opposite poles. Here we can see that we are seeing the exchange of genetic material taking place. So this, we can say that it is prophase 1, right? And then here we have prophase 2. And then we also have prophase 1 here. Right. Okay. Now let's have a look at the questions. You're going to go through the questions for a few minutes, and then we will discuss the questions later. Okay, let's have a look at the questions. The first one is 2.4.1. It says identify the type of cell division represented above. Right, so we know that it is meiosis. However, is this meiosis 1 or meiosis 2? So here we are looking at meiosis 1. Why? Because we can see crossing over taking place. In meiosis 2, crossing over does not take place. What is another reason? Chromosomes are represented in homologous pairs. We do not see chromosomes in homologous pairs when we are looking at meiosis 2, right? And then the number is also reduced at some point. At the end of the phase, numbers of chromosomes are reduced. Remember, at the end of meiosis 2, the chromosome numbers do not change, right? And then identify phase E. Let's go back. That is prophase 1, right? Because we can see crossing over taking place, right? So that is prophase 1. 2.4.3, use only the letters A to E to place the phases shown above in the correct sequence. So remember, we are going to look at from the first phase until the last one, which is telophase, and then we will look at that. Here, it is going to be C, E, B, A, D. And then for this kind of a question, even if it's not a question like this, but you are asked to give the letters that represent this kind of a question, make sure that you put them in the correct order, because if you just miss one, you're not going to get half marks. You're going to lose all the marks. So you either get 2 marks or you do not get anything. So C, E, B, A, D, meaning that you are going to place them in the correct order, which is going to be interphase here, not prophase, right? And then we are going to have E, which is prophase, right? Prophase 1, right? And then we are going to have anaphase, and then telophase, and then this one would then be prophase 2, right? So this would be in meiosis 2, basically. All right, now let's move on to the next one. 2.4.4, describe how a chromosome mutation may lead to trisomy 21. Right, when we are talking about chromosome mutations, we would be talking about things like non-disjunction. That non-disjunction leads or is a representation of a chromosomal mutation, because there was a mutation in the way the chromosomal structure is supposed to be, right? So you would mention that during anaphase, either 1 or 2, non-disjunction of the chromosomes pair 21 would occur, right? Obviously, because it said trisomy 21, you are going to make sure that you talk about the chromosomes being extra. There's going to be extra chromosomes on pair 21, right? And that chromosome that has an extra pair on chromosome 21 will then fuse with a normal gamete. I mean, that gamete with at 21 would have an extra chromosome, and it will fuse with a normal one, and that normal gamete would have haploid number of chromosomes, and then it will lead to a zygote with 3 chromosomes on number 21. And we all know that that would then lead to a Down syndrome, right? And then we also have polyploidy, right? So with polyploidy, remember in Down syndrome, we're looking at aneuploidy, right? So here, this is when there is non-disjunction of all the chromosomes during meiosis. So it's not whereby some chromosomes fail to separate and some chromosomes separate. Here you are going to be experiencing that all the chromosomes have failed to separate. Therefore, the gamete will then have an extra set of chromosomes, not just one chromosome, right? Whereas the other one will have no chromosomes at all, right? So if that gamete with the extra chromosomes fuses with a normal gamete, it means that now we are ending up with 3N. Remember, if it's just a chromosome, you would have 2N plus 1 at the end of fertilization. So in this case, because we have a normal gamete, which is N, plus a gamete or crossed with a gamete that has 2N, because all of them have failed to separate, it means that the cell remains diploid even at the end of meiosis. So N plus 2N, this is normal. This is the abnormal one that failed to separate. So when that happens, you are going to result in 3N, which is a triploid cell, right? So we can see here, these two have failed to separate. These two chromosomes have failed to separate. So we have a diploid cell with a cell that has 0 chromosomes, right? So when that happens, because all the chromosomes have failed to separate, we are then going to see monosomy in one cell and polyploidy in the other cell, right? So we also have tetraploid organisms whereby an abnormal gamete fuses with another abnormal gamete. So here we start off with this. So this is at the beginning, right? At the beginning, we have a diploid cell. Now this diploid cell fails to separate. At the end of meiosis, you still end up with a diploid cell. At the end of meiosis, again, you still end up with a diploid cell. When these two diploid cells, these two abnormal diploid cells, when they fail to separate and they fuse with each other, it means it's going to be 2N multiplied by 2N, or 2N plus 2N, giving us 4N, right? So that is referred to as tetraploid. So we have diploid, we have haploid, diploid, triploid, and tetraploid. So tetraploid would be a result of 2 gametes that are both abnormal and all the chromosomes failed to separate, right? So this can also take place in plants, whereby plants or animals are polyploid and they have more than 2 sets of chromosomes, right? It is, however, more common in plants, and it can actually be an advantage if this happens in plants. In animals, it can happen whereby we can see that we have a tetraploid rat that was discovered, and we can see that it is actually of a much bigger size than usual. Whereas in plants, when polyploidy occurs, especially in agriculture, you would then find that it is an advantage because the plants would contain bigger fruits. So these plants will bear big fruits, and those would then become fruits that are actually more desirable. Right. So the advantage there would be the fact that it would form seedless varieties. Sometimes you find that there are seedless varieties in watermelons and bananas as well as apples, or the cells are bigger. Therefore, the flowers, the fruits, and the storage organs are also bigger. And then it can also cause some infertile plants to become more fertile. And then it can also cause these plants to be disease resistant, meaning that they can become healthier. Then it means that the chances of survival would increase. That is more yield for farmers in agriculture, right? So a normal fruit would be this size as we normally know it. As we can see here, we have 1, 2, 3 chromosomes. However, here we have 1, 2, 3, 4, 5, 6. It means that here they've all failed to separate, leading to a polyploid fruit. Right. Now let's move on to the last part, which is the difference between mitosis and meiosis. So remember, here, during mitosis, crossing over does not take place. Mitosis does not experience crossing over. Crossing over only happens during prophase of meiosis 1. Why does crossing over not take place during mitosis? We don't need the cells produced to be different from each other. The cells produced by mitosis are the same. Therefore, you would see that the cells are going to be the same at the end of mitosis. You can see that crossing over is not taking place there. And then in anaphase, when we look at anaphase, we can see that whole chromosomes are pulled to the opposite poles, right, in anaphase 1, whereas in mitosis, we do not have two divisions. We do not have mitosis 1 and mitosis 2, only mitosis. So during mitosis, it is the chromatids that are going to be pulled to the opposite poles, which then takes us back to that thing whereby we say mitosis is similar to meiosis 2. Because in meiosis 2, it's chromatids that separate. So this would be a representation of mitosis. As we can see, the chromatids are being pulled to the opposite poles. Whereas in anaphase 1, it's whole chromosomes that are being pulled to the opposite poles. Now let's have a look at the other differences. 2 nuclear divisions in meiosis. Meiosis 1 and 2, only 1 nuclear division in mitosis. 1 cell divides to form 4 cells, whereas mitosis only forms 2 cells. And then the chromosome number is halved. This leads to the cells being different, whereas in mitosis, the chromosome number remains the same. They have to remain the same for us to be able to say the cells are the same. And then 4 new cells that are formed are genetically different from each other due to crossing over. These cells are the same because crossing over did not take place. And then the next one, this usually results in gametes, formation of gametes in meiosis, whereas in mitosis we form somatic cells. Remember, the somatic cells have to be the same, so that if you take any genetic material from a human body or the body of an organism, you will find that the material is the same because they were formed by mitosis. Crossing over occurs in prophase 1, whereas crossing over does not occur during mitosis. And lastly, during anaphase, whole chromosomes are pulled to the opposite poles, whereas the chromatids are being pulled to the opposite poles during mitosis. Okay. And then the similarities, we have a DNA replication, which takes place during interphase for both mitosis and meiosis. And then the nucleus divides at telophase, whereby we are going to see the 2 new cells being formed at the end of mitosis and at the end of meiosis 1. And then also each cell will experience the same at the end of meiosis 2. The cytoplasm also divides during both phases, and this leads to the formation of new cells, which can also occur in both plants and animals. Let's have a look at our last activity. 3.4, it says scientists conducted a survey to establish the relationship between the age of the mother and the risk of her having a baby with Down syndrome. They recorded the number of babies born with Down syndrome per 5,000 births over a 1-year period and recorded the age of the mother. Their findings are summarized in the table below. We have age of the mother in years, and we have the number of babies born with Down syndrome per 5,000 births. So if the mother is 25 years old, within those 5,000 births, only 10 had Down syndrome. And then as the age increased, at 35, about 15 babies were born with Down syndrome out of 5,000 babies. At the age of 45, the numbers increased up to 300 babies with Down syndrome out of 5,000 births. Okay, just a few minutes to go through the questions. Remember, 1 minute, 1 mark, meaning that you are going to be given a few minutes just to give an attempt to these questions before we look at the answers. Okay, let's have a look at 3.4.1. Which age of mother has the highest risk of having a baby with Down syndrome? As we can see here, we have the ages 25, 35, and 45. And the age 45 is the one with the highest risk. Okay. And then 3.4.2. It says describe 4 planning steps for this survey. So you need to plan when you when you are about to conduct a survey, right? So for you to get the results, you need to consult with hospitals, right, or maternity homes in order to get permission to gather the information about their patients, right? So you need to get the permission from the hospital and also get permission from the mothers to use their data. Right, you cannot just take information about individuals without their permission. Then also, the scientists will determine how large the sample should be. Sample size. We know that when we look at investigation, sample sizes are very important. If we do this using only 10 births per year, it will not give us the same reflection of results as we do with 1,000 births or with 5,000 births. So that is also very important. Make sure that the sample includes expectant mothers of various ages, which is what was done here. Or we can have even more different ages. So from 25, 30, 35, 40 until we reach 45 or even more. And then decide how the results will be recorded and the time period where you'll gather the results. In this case, it was 1 year. 3.4.3, state the independent variable and the dependent variable. So remember, the age of the mother is the one that has an effect on whether the child is born with Down syndrome or not. So it means that the age of the mother is independent, and whether the child is born with Down syndrome or not is the dependent variable. So independent variable, age of the mother; dependent variable, the number of babies with Down syndrome. 3.4.4, state one way of improving the reliability. Remember here, when we talk about reliability, always reliability, we are talking about repeating the investigation and increasing the sample size. So if we use 5,000 births, we can increase that and use more. So increase the sample size or just do the investigation again and then get the average. So conducting the survey over more than 1 year, that is repeating the survey. Okay. 3.4.5, state a conclusion for the survey. Obviously, from the results that we saw, it's that as the age of the mother increases, the chances of, or the numbers of having babies, the numbers of babies born with Down syndrome also increased. Or you can say that the younger the mother, the numbers of babies with Down syndrome were lower. So your conclusion can speak about the mothers that are older or the mothers that are younger. So the older the mother, the higher the risk of having a baby with Down syndrome. So now, just to summarize what we went through, the importance of meiosis, 4 things here. Production of gametes, right? We need gametes here. reduction of chromosome number. That is where we talk about the chromosome numbers being constant by introducing the halving effect to avoid the doubling effect of fertilization. And then, okay, that is the maintenance of chromosome numbers. Lastly, variation, genetic variation. What introduces genetic variation? Crossing over and the random arrangement of chromosomes at metaphase. And then with mitosis and meiosis, on one side we have meiosis and the other mitosis. These are the differences. And then in the center we have the similarities. We just discussed these, whereby we know that we can look at meiosis taking place certain ways and mitosis taking place a certain way as well. We know that, for example, it involves one cell division mitosis, and meiosis involves two cell divisions, right? Meiosis produces 4 haploid cells, whereas mitosis produces two diploid cells that are genetically identical. Right, so now in the next lesson, we will be looking at diversity of reproductive strategies. So here we are going to be looking at internal fertilization, external fertilization. We're going to be looking at ovipary, vivipary, as well as ovovivipary development. Right, okay, we have come to the end of our lesson for today. Thank you for watching. I will see you next time. Goodbye.